Today I'm going to introduce to you a technique that we've been working on in our lab for several years now. And it essentially tries to make the use of, let's call it spatially resolved omics in single cell context routine. So I think all of us in this room kind of recognize the importance of spatially resolved transcriptomics and would like to extend some of these techniques like we saw in the talk from Miguel Esteban earlier to the proteomic level as well. And we've really seen a number of methods that allow us to gain spatial information at the RNA level. Many of them use single molecule fish or derivatives of single molecule fish to sort of tile DNA probes along an RNA and then either do some sort of amplification to increase the signal that makes them less quantitative, or in the case of merfish, use very sophisticated sequential probing to be able to reveal multiple probes, multiple genes at the level of a single cell. In fact, with techniques like merfish, you can go up to 1,000 transcripts in a single cell, and really the limitations are related to degradation of the actual sample. We also have some really far out techniques that nobody's mentioned today, like TomoSeq, and which basically tries to use some variants of tomography to image cells at different angles, and also TivaSeq, which tries to do this in an in vivo analytical approach. But the question is also, why are we doing this? Why are we so interested in being able to recover this sort of spatial information? And really, it's because we have a basic recognition that messenger RNAs, which go into the cytoplasm, are somehow related to the local positioning of proteins in their second parts of their biological life. And we know that in some cases, the RNA-protein relationship is direct, so you make an RNA, it transits into the cytoplasm, and the protein is made locally where the RNA is, or the RNA is made in one place, goes into the cytoplasm, makes a protein, and that protein itself is what traffics to its final destination. But intrinsically, I think this question came up for me when I was working a lot on fly development, and here we can see a protein in green, which is called Staufen, and Oscar, which is a messenger RNA, which is made in this cell and then seems to traffic into what will become that seems to traffic into what will become the future embryo into this compartment. And being able to sort of understand this change in RNA positioning and the direction of this Oscar RNA to the posterior here is important, because that in turn results in the formation of the posterior part of this egg. And so using this kind of probe technology of molecular beacons that was developed in our PhD lab, we can really understand in time where these things occur, and in a way we can reveal cellular state. And I think that's a really big question for us in this field, is that even though we use destructive techniques like single cell RNA sequencing, and we try to create pseudotime computationally, our real problem is that biology takes place in 3D, and we really cannot easily relate cell state from these snapshots of RNA to the spatial and temporal outcomes of where this RNA is. And we see that really clearly in different examples here, for example, in neurons, or in cell division and development, or for example here, which is a really interesting case study where you have the change in the positioning of this RNA, ApoB, depending on the grandial state of this mouse. So to make that less complicated, depending on when this mouse eats, this RNA, ApoB mRNA, changes its position from the apical to the basal position. And so counting the number of RNAs uniquely here doesn't tell you actually what's occurring in this enterocyte, in this cell state, because it's just flipping from one position to another. And of course, we know this from long ago from the work from Rob Singer's lab and Edouard Bertrand when they looked at ASH1 messenger RNA, or in hippocampal neurons, as I mentioned earlier. So our laboratory has worked really hard to try to come up with a combination of techniques and computational methods that can sort of reveal to us the dynamic cell state and RNA protein relationships that exist at single cell resolution. So in an attempt to do this, we relied on a previously existing technique of micropatterning. And really, because again, biology takes place in 3D, micropatterns attempt to recreate these microenvironments and geometries that naturally exist in biology, but they superimpose them using sort of microfabricated surfaces that allow cells to adhere into these geometries. And when you do that, you can see these really beautiful stainings you can see with actin and microtubules. And sort of by being able to do that routinely, we can image hundreds of cells and we can position them in different geometries on these cells and put them in contact with each other to elicit specific physiological phenotypes. Now, one of the things that became revealed in this sort of work is that we had reproducible positioning of proteins on micropatterned cells, suggesting that cells contain an inner GPS that positions proteins and cellular organelles reliably to the same positions. And that could be quantified, and this is from Bruno Goud's lab at the Institut Curie in Paris, where you can see that there's enrichment of specific regions of the cell in these micropattern cells for these specific vesicular and trafficking proteins. So the real question is, can we use a similar technique to reveal this GPS map that may or may not exist for RNAs? And so we've attempted to do that by patterning cells and then revealing the positions of specific messenger RNAs, which by using single molecule FISH and specific pipelines where we can use image processing to denoise and fit and align multiple cells to the same geometries, and then by volume segmentation, look at them in 3D, be able to reliably superimpose thousands of cells on top of each other to look at the positions of RNA and proteins and see how reproducibly a specific RNA and protein go to a specific place. And you'll see in a moment that that alone isn't enough, because we still need to apply some different algorithms and mathematical filters to be able to reveal clustering or non-clustering or non-positioning of these RNAs and proteins. So this is just showing how we initially microfabricated these techniques, but importantly, when you do this, you see that the deviation in the position of these central dogma molecules becomes very constant after a low number of cells. So after about 30 cells, we see that we have very little deviation in the positioning of RNA and protein, suggesting that when cells assume specific geometries, whether they're epithelial cells or highly organized cells, they reproducibly position molecules in the same place. So when we did this, we could do this for multiple RNAs and proteins, and we could see that by induction of transcription or mobility in migrating cells, we could actually see specific transcripts that would localize at the periphery more richly than they would, say, in the center of the cell. And we could fit those to an algorithm, a Ripley-K algorithm, which has two biases, one for complete spatial randomness and another one that showed them as spatially clustered. And when we fitted these over time, we could see that first we could see an RNA peak, and then we could see a subsequent protein peak in each of these different cases, whereas for some genes like GAPDH, we would see a fit to complete spatial randomness. Now, what's really important is that the MTOC marks the center of the cell, and we could actually position this sort of organelle to how it would be influential to the directionality of that positioning. So here you can see that we derive a MTOC polarity index, and the MTOC polarity index is deterministic of where the RNA of this protein, of this gene goes, but not where the protein goes. Likewise, it's highly deterministic early for part D3, but less so in later times, and more deterministic for the protein at late times. And here we're following the position of these RNAs over time by doing FISH and single molecule FISH and immunofluorescence. So we could use this type of analysis together with spatial analysis at different positions of the cell to assess specific areas where we have enrichment at different time points, and also superimpose this on where we see the MTOC being deterministic of where those enrichments happen. So sometimes those enrichments happen in the same compartment as the MTOC or same directionality as the MTOC, and other times the MTOC is in a completely different compartment from where the enrichment of the RNA is occurring. So the last thing I'll show you in the last 10 seconds I have is that we can apply this to very large cells like muscles, where you can see that we have the same number of molecules of this actinin and 2 messenger RNA that seem to distribute in the immature fiber, but later seem to be in these same striations. So this is a classic example of changes in cell state, and the clustering that we see here is actually related to the formation in the mature muscle of these Z lines. So this is kind of an outstanding question in the field, and then we could use our Dypfish algorithm to look actually at the position of actinin and 2 messenger RNA. And this is its position in the immature muscle fiber, which seems to have absolutely no organization. But as time progresses, it starts to resolve into these discrete striations or lines where the mature muscle fiber protein actinin 2 correlates to the position of this actinin 2 messenger RNA. And now that's emerging from the 2 nuclei. This is a very large cell. That if we did single cell RNA-seq, we wouldn't actually be able to see this because the number of RNA molecules is essentially the same in these 2 different conditions. And we control that with GAP-DH. So in the future, what we see here is that we need technologies that allow us to do this at very high bandwidth. And high bandwidth means like in tissues, but with single cell resolution, which is something that we're working on extensively in our laboratory. So with that, I just want to summarize and thank the organizers. I also want to thank the people who we worked on this project. I don't know why it's not coming, which was really run by Anke Savlescu in our lab and Robin Bracken, and we collaborated with Masha Nikolsky's lab and Edgar Gomez's lab in France and also in Portugal. And this work is supported by the Chan Zuckerberg Initiative.